Recovery of Mendel's work after 16 years of his death in 1900 by three researchers independently: Karl Correns, Germany; Hugo de Vries, Holland; and Schermack, Austria. Karl Correns, Germany; Hugo de Vries, Schermack, and Hugo de Vries, Holland; Schermack, Austria. Karl Correns, Germany; Hugo de Vries, Holland; Schermack, Austria. He could not provide physical proof for his factors or scientific work. Was busy in studying Darwin's work, limited circulation of the journal, and communication was not easy, so his work was not published. Use of mathematical tools and statistical analysis was new to biologists. He discarded theory of built-in inheritance is unacceptable by scientists of his time. By scientists of his time. So, reason for his success, Mendel considered one or two characters at one time. Mendel considered one or two characters at one time. Use of statistical methods and uh, mathematical logics for analyzing his results. Large sampling size kept complete record of every cross, studied inheritance till F3 generation, studied inheritance till F3 generation. His choice of experimental model, garden pea plant. His choice of experimental model, garden pea plant. So, the reason for failure of Mendel, scientific world was busy in studying Darwin's work, circulation limited circulation of the journal, and communication was not easy, so his work was not published. Communication was not easy, so his work was not published. And so his work was not published. Use of mathematical tools and statistical analysis was new to biologists. Use of mathematical tools and statistical analysis was new to biologists. He discarded the theory of blending inheritance was unacceptable by scientists of his time. And sixth, he could not provide physical proof of his factor. He could not provide physical proof for his factor. Reason for his success, Mendel considered one or two characters at one time. Use of statistical methods and mathematical logics for analyzing his results. Large sampling size kept complete record of every cross and kept complete record of every cross. Studied inheritance till F3 generation. Studied inheritance till F3 generation. Oh, dude. Hi. Structure. Studied Studied inheritance till F3 generation. His choice of experimental model, garden pea plant. Mendel considered one or two Mendel considered one or two character at Mendel considered one or two character at one time. Use of statistical methods and mathematical logics for analyzing his results. Large sampling size kept complete record of every cross. His choice of experimental model, garden pea plant. His choice of experimental model, garden pea plant. So reason for his success, Mendel considered one or two characters at a time. Use of statistical methods, use of statistical methods and mathematical logics for analyzing his results. Next হচ্ছে large sample, large sampling size. Fourth, he kept record of every cross studied inheritance till F3 generation. He kept record of every cross studied inheritance till F3 generation. And last, his choice of experimental model, garden pea plant. His choice of experimental model, garden pea plant. Mendel selected garden pea because of its short life cycle, easy to grow, rate of seed production is high, observable, many observable, detectable contrasting traits of characters, bisexual flowers, hence cross, self and cross, both pollinations possible. So Mendel selected garden pea because short life cycle, easy to grow, short life cycle, easy to grow. Rate of seed production is high, short life cycle, easy to grow, rate of seed production is high, and many observable, detectable, contrasting traits of characters, many observable, detectable, contrasting traits of characters, bisexual flowers, hence self and cross, both pollinations possible, bisexual flowers, hence self and cross, both pollinations possible. Technique used by Mendel, parents, pure line, example, pure line tall, male, or pure line dwarf, female. First is the গাছের emasculation removal of stamens in bud stage to prevent self-pollination. Uh, the cross-pollination করানোর জন্য parents pure line ছিল technique used by Mendel. Parents first pure line ছিল pure line tall, pure line dwarf. Now, first is emasculation, then bagging, debagging, then cross pollination, then rebagging. So, emasculation, removal of stamens in bud stage to prevent self pollination. Emasculation, removal of stamens in bud stage to prevent self pollination. Emasculation, removal of stamens in bud stage, removal of stamen 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 in bud stage to prevent self-pollination, removal of stamen in bud stage to prevent self-pollination, removal of stamen in bud stage to prevent self-pollination, removal of wait, 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 wait. Emasculation, removal of stamens in bud stage to prevent self-pollination. Then bagging করা হয়েছে to prevent cross-pollination with undesired pollen grain. To prevent cross-pollination with undesired pollen grain. Fourth হচ্ছে then debagging করে dusting of desired pollen grain on stigma, dusting of desired pollen grain on stigma, dusting of desired pollen on stigma, dusting of desired pollen grain 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 on stigma, of desired pollen grain on stigma, dusting of desired pollen grain on stigma, thereby rebagging. So, emasculation, removal of stamen at bud stage to prevent self pollination, then তারপর করা হচ্ছে bagging to prevent cross pollination with undesired pollen grain. Third হচ্ছে debagging. Fourth, dusting of desired pollen grain on stigma. Fifth, rebagging. This technique is called emasculation and bagging. This technique is called emasculation and bagging. So F1 seed sow করা হলে then এরপরে F1 seedকে sow করা হবে F1 plant এই কনভার্ট হওয়ার পর F1 seedকে sow করা হলে F1 প্লান্টে যখন F1 plant তৈরি হবে F1 প্লান্টে সেলফিং করানো হবে দেন F2 seed তৈরি হবে না F2 seedকেও sow করা হবে F2 প্লান্টে সেলফিং করানো হবে পরে F3 জেনারেশন এমন করে till F3 জেনারেশন নিয়ে এটা স্টাডি করা হয়েছে Phylum Sativum deployed 14 to in 14 Mendel considered 7 characters so 14 traits and 14 pure lines 14 pure lines Mendel considered 7 characters মানে 14 traits আর 14 ই হচ্ছে pure lines Mendel considered 7 characters 14 traits Mendel considered 7 characters 14 traits 14 pure lines dominant trait or recessive trait chromosome number stem or plant height stem height or plant height chromosome number 7 stem height or plant height chromosome 7 capital T small t stem height or plant height stem height or plant height chromosome number 7 dominant trait capital T recessive trait is small t tolerance broadness stem or plant height chromosome 4 stem or plant height chromosome 4 T দিয়ে এটা ডিনোট করা হচ্ছে seed color yellow dominant recessive green seed color yellow dominant recessive green chromosome number 1 seed color chromosome number 1 seed color chromosome number 1 seed shape round capital R wrinkles smaller chromosome 7 seed shape chromosome 7 seed shape chromosome 7 flower color violet dominant or white recessive violet dominant recessive white into chromosome number 1 so flower color seed color chromosome 1a seed color flower color chromosome 1a flower position axial or dominant terminal recessive axial dominant terminal recessive chromosome number 4 pod or green dominant yellow recessive chromosome number 5 pod or fruit shape full or inflated eye are constricted but wrinkle constricted small eye recessive chromosome number 4 so height of plant chromosome 4 height of plant chromosome 4 the chromosome 4 এর কি আছে height of plant flower position height of plant flower position or fruit shape chromosome 4 height of plant chromosome 4 height of plant flower position or fruit shape height of plant chromosome 4 height of plant flower position fruit shape chromosome 4 height of plant flower position fruit shape height of plant chromosome 4 fruit shape chromosome 4 flower position chromosome 4 height of plant flower position fruit shape chromosome 4 chromosome 1 has a seed color and flower color seed color and flower color chromosome 1 seed color flower color chromosome 1 seed color flower color chromosome 1 seed color flower color chromosome 1 seed shape chromosome 7 seed shape chromosome 7 আর fruit color chromosome 5 seed shape chromosome 7, fruit color chromosome 5, seed shape chromosome 7, fruit color chromosome 5, seed shape chromosome 7, fruit color chromosome 5, seed shape chromosome 7, seed shape chromosome 7, fruit color chromosome 5, seed shape chromosome 7, fruit color chromosome 5. So, stem height or plant height, stem height or plant height chromosome 4, seed color chromosome 1, seed shape chromosome 7, flower color chromosome 1, flower position chromosome 4, fruit color chromosome 5, fruit shape chromosome 4. Inheritance of one gene, monohybrid cross. Inheritance of one gene, actor gene inherit monohybrid cross. Inheritance of one gene, 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 monohybrid cross. Acta gene, mono, acta gene. Inheritance of one gene, monohybrid cross है. So study of inheritance of single pair of contrasting traits of a character at a time. So inheritance of one gene basically monohybrid cross. The study of inheritance of single pair of study of inheritance of single pair of contrasting traits of a character at a time. The inheritance of one gene monohybrid cross. Study of inheritance of single pair of contrasting traits, single pair of contrasting traits of a character at a time, like pure line tall, pure line dog, pure line tall, capital T, pure line dog, small t, এর cross করানো হলে all first তো all tall পাওয়া যাবে, capital T small t, capital T small t, all tall, সেখানে selfing করানো capital T small t আর দুটো একটা ক্যাপিটাল টি একটা স্মল টি মানে একটা পিওর টল একটা পিওর ডর আর আর দুটো ক্যাপিটাল টি স্মল টি পাওয়া যায় ওরাও টল হয় সো এটা ছিল F1 all were tall none were dwarf trait of one parent present so একটাই প্যারেন্টের ট্রেট টলটা প্রেজেন্ট রয়েছে trait of other parent is absent এটা হচ্ছে ক্যাপিটাল টি স্মল টি হচ্ছে গ্যামেট now এটা F2 all resembled one of the parent, no intermediate. So trait 1 F1 all were tall, trait F1 all were tall, none were dwarf, trait one parent present, trait of other parent absent, all resembles one of the parent, no intermediate. Can you move? Can you? Okay. Yes, so this all resembled one of the parent, no intermediate. All resembled one of the parent, no intermediate. আর F2 তে কি হচ্ছে both tall and dwarf দুটোই আছে একটা dwarf তিনটে tall so both tall and dwarf দুটোই আছে in ratio 3:1 so 75% tall and 25% dwarf resembled both parents so F2 resembled both parents no intermediate form seen still no intermediate form seen হয় tall নয় dwarf so 3:1 ratio তে F2 তে so law of dominance, each character is controlled by a discrete unit called factor. Now we call it call now we call it gene, which is the unit of inheritance contain information for expression of a trait, contain information for expression of a trait. Law of dominance. Each character is controlled by a discrete unit called factor. Each character is controlled by a discrete unit called factor. Each character is controlled by a discrete unit called factor. Law of dominance. Each character is controlled by a discrete unit called factor. Now, now we call it gene, which is the unit of inheritance contain information for expression of a trait. Uh, Now these factors occur in pair, মানে one gene, দুটো allele থাকে, two alleles, one gene, two alleles. Factor occurs in pair. These factors they occur in pair, one gene, two alleles. If two alleles are dissimilar, example capital T small t, then one dominates the other. If two alleles are dissimilar, example capital T small t, then one dominates the other. জিনোটাইপ ক্যাপিটাল টি স্মল টি আর ফিনোটাইপ হচ্ছে টলনেস, ওই টলনেসটা হচ্ছে ফিনোটাইপ, আর ওর জিনোটাইপ যেমন ক্যাপিটাল টি জিন স্মল টি এটা হচ্ছে জিনোটাইপ। ক্যাপিটাল টি হচ্ছে ডমিনেন্ট অ্যালিল আর স্মল টি ইজ রিসেসিভ অ্যালিল। ক্যাপিটাল টি ডমিনেন্ট অ্যালিল, স্মল টি রিসেসিভ অ্যালিল। So law of dominance explains expression of only one type of parental trait in F1, expression of both parental trait in F2, proportion of 3:1 in F2 generation. Law of dominance is proven by monohybrid cross. So law of dominance is proven by monohybrid cross. Law of dominance explains expression of only one type of parental trait in F1, expression of only one type of parental trait in F1, expression of only one type of parental trait in F1, expression of both type of parental trait in F2, proportion of 3:1 in F2 generation, 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 proportion of 3:1 in F2 generation. Law of dominance is proven by monohybrid cross. Law of dominance is proven by monohybrid cross. Law of dominance is proven by monohybrid cross. So parent capital T, small t, মানে pure line tall, pure line dwarf. তাদের gametes হবে capital T and small t. F1 এ all capital T small t all tall, F1 এর selfing করালে F2 তে gametes ও থাকবে capital T দুটো, দুটো small t, ওখানে F2 তে selfing করালে capital T capital T capital T small t capital T small t and small t small t. It is shown tilted in NCERT. The Punnett square. So F2 generation এ phenotype ratio হচ্ছে 3:1, তিনটে dominant একটা recessive obviously, আর genotype ratio হচ্ছে capital T 1, capital T small t দুটো and small t একটা, so 1:2:1. Genotype ratio 1:2:1. Genotype is small detail. আর জিনোটাইপের মধ্যে একটা হচ্ছে হোমোজাইগাস ডমিনেন্ট, একটা হোমোজাইগাস রিসেসিভ আর একটা হেটেরোজাইগাস। সো হোমোজাইগাস ডমিনেন্ট, heterozygous and homozygous recessive. Punnett square. Mendel proposed that something is being stably or in unchanged form passes from parent to offspring through gametes over successive generation, and he called it factor. With Punnett square. And Mendel proposed that something is being very stably or in unchanged form passes from parent to offspring through gametes over successive generation. He called it factors. So after law of dominance, okay. Mendel proposed that something is